Okay, so let's start. This is week 3 of Biophotonics, and I'm Brigrishi Mishra. In this week 3, mostly basics of biology has been taken into consideration. So, like, I'm not a biology expert, I don't have a biology background, so I don't know much about this field. So that is why this is gonna be a very short session, as we also don't have much numerical problems to solve. Not much, I mean, not even a single numerical problem, it's just basic theory questions. So I'll just discuss the questions given in the assignment. Okay, and I mean, of course, you know the answer, so we'll just discuss uh, basic things, and then we will end this session. And of course, if there are biology students, please correct me if I'm wrong. I mean, if I do any mistakes, or if I make any wrong statement, please correct me, because I also don't know anything about the biology. Whatever I studied in 10th standard, that's all. So I have prepared the contents for this class based on the Wikipedia. Okay, so most of the statements I'll be stating will be available on the Wikipedia page. Okay, so just correct me if I make any wrong statement. So the first question is, nucleoside contains base sugar, base phosphate, base sugar phosphate, and sugar phosphate. So four options are given. So the correct option is base sugar. Okay. Why is it so? Because so the first thing is, what is nucleoside? Okay. So it's basically it contains nucleobase and 5 carbon sugar. So the, the the basic definition I found about nucleoside on the Wikipedia is that nucleoside sorry uh, nucleoside can be thought of uh, as nucleotides without a phosphate group. Okay, without a phosphate group. So clearly it doesn't have phosphate. Okay, so in all these four options, only this first option doesn't have phosphate. Okay, other three options, base phosphate, base sugar phosphate, have the phosphate group. Okay, so the answer is base sugar. Another thing is, so it is like it contains nucleobase. So what is nucleobase? That is an important thing. Okay, so nucleobase. Okay, so these are nothing but simply bases. Okay, these are simply bases. What kind of bases? So these are nitrogen containing biological compound. Nitrogen containing biological compounds. Okay, so why nucleobase are important? Because like they form base pairs. Okay, and they are responsible for this helical structures of RNA and DNA. So they form base pairs, and to stack one upon another leads directly to long chain helical structures such as RNA and DNA. Okay, so and there are five nucleobases. So five nucleobases. What are these? Adenine, which is represented as A, cytosine represented as C, guanine represented as G, uh, thymine represented as T, and uracil, so it's G, oh sorry, and uracil is represented as U. Okay, so this comes to our next question: bases which are present in RNA but not in DNA. So these are the five nucleobases we have. Okay, A, C, G, T, U. So these five are present. So the combination of these five, okay, present in RNA and as well as in DNA. So in RNA, this uracil is present in place of thymine. Okay, so others are same. So for RNA, adenine, cytosine, guanine and uracil is present. Okay, and for DNA, adenine, cytosine, guanine and thymine is present. So the right answer is I think C. Okay, yeah, the right option is C, because it doesn't have thymine, okay, which is present in the DNA, not in RNA. So which organism was used in Griffith's experiment to prove that genetic materials can be transferred through a process called transformation? Okay, so it's that rat experiment where a strain is injected into mice, and the mice died. When the R strain injected into mice, it lived, and then the further process. So that process was called transformation process. So the organism which was used in that process was this Streptococcus pneumonia. Actually, Griffith was doing the experiment. It was, I think, post World War First. He was in a search of vaccine of pneumonia. Okay, because at that time pneumonia was a very deadly disease and a lot of people was dying at that time. So he was just like he was in search of creating a vaccine for pneumonia. Okay, so Griffith's experiment suggests that bacteria are calculator here of transferring genetic information through a process known as transformation. So basically what sir told in the video, that is the basis of this DNA RNA, like how they have this information, okay, transferring from one cell to another cell and so on. Okay, so next question is, which cell organelle is mainly responsible for the process known as translation? So first, what is translation? Okay, so the definition of translation is, so it is the process in which ribosomes. Okay, it is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. Okay, and this entire process is called gene expression. So clearly it is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. So it is the ribosomes, okay, which is mainly responsible for the process known as translation. What is the nucleotide sequence of start codon? Okay, so uh, so what is start codon? So first, what is codon? Okay, so codon is a sequence of 3 nucleotides. It is sequence of 3 nucleotides which together form a unit of genetic code. Okay, so codon are sequence of 3 nucleotides which together form a unit of genetic code, okay, in either DNA or RNA molecule in a DNA or RNA molecule. Okay, so they basically indicates whether the translation should begin or cease. So in case of start codon, okay, so start codon is the first codon of a messenger RNA transcript translated by a ribosome. So the in RNA the start codon is AUG. So the correct option is AUG. Also, when it is happening in DNA, then in DNA we don't have uracil, so it is ATG, that means thymine. So the start, or I should write just, so it is ATG. Okay, so the start codon for DNA is ATG, and for RNA it is AUG. Who got the Nobel Prize for the work of CRISPR technology? So the correct option is Jennifer Doudna. So I mean, you have heard the interesting story of this CRISPR technology and how like there were 3 professors who were working simultaneously but in the different part of world on this technology, and the 2 got the Nobel Prize, but the one didn't, just because his paper wasn't accepted by the by the uh, by the reviewers or editors. Okay, because they didn't believe in his work. So yeah, that happens in the research community. even sometimes researchers don't publish their work just because they think that how is this possible. It is wrong. I have I I probably have made some mistake because this cannot happen. Like for example, the Planck's constant E equals h nu. Okay, so uh, like if you read there, you will find lot of incidents in the scientific history. Okay, so what is the connective tissue in human body? So it is of course blood, because the other three are not the connective tissues. So what is connective tissue? I just want to write a definition of connective tissue. So connective tissue, so it supports and connect different tissues and organs of the body. Okay, so it supports and connect different tissues and organs of the body. So clearly it is blood who does all these things. It contains three types of fibers. Contains three types of fibers: collagen, elastic, and reticulous. Okay, the yeah. So the next question is disease that cause for protein misfolding. So protein misfolding is where rather than going in the exact or the correct shape, there is some misfolding and it creates another structure. Okay, so that appears in form of disease, and that can cause several disease. So whatever the mentions here, Brian, Alzheimer, Parkinson, all are due to the protein misfolding. Okay, so the correct option is all of them. Name of the receptor of coronavirus uses to enter the host cell. So when I search this question on the Google, so this name was suggested, ACE2. Okay, so but when I read a paper, I got to know that this is the correct answer, spike. So I just want to write the definition of spike. So the spike S. So spike is basically a protein, okay, covered by the lipids. The spike means in this process the spike is covered by the lipids. Okay, in this coronavirus thing. So the spike S protein, the spike protein of of COVID, yeah, the spike protein of coronavirus mediates the recognition of the host cell receptors and facilitates the cell attachment and the cell membrane fusion during the viral infection. Okay, and this ACE2 is a component of spike. So it has spike S. So it has two components, S1 and S2, and AC2 is a component of the any of these S1 and S2. Okay, so it is the spike protein which is the receptor of coronavirus uses to enter the host cell. The last question is, what is the function of lipids in cellular structure? So, what are lipids? So, lipids are a group of compounds in living systems that are soluble in non-polar solvents. So non-polar solvents like hexane, which is hydrocarbon C6H12. Okay, so these are soluble in non-polar solvents. So it is stored in the body as a fat and serves as an energy reservoir that is metabolized when needed, and it also gives structural stability. So the right answer is both A and B. Okay, uh, so yeah, that's it today from my side. I don't have much to teach of biology. Okay, but if you want to know the basics, either you can like first I'll suggest you to listen the lectures of week 3, and then you can just if you have any doubts, you can search some search on the Google. You will find a lot of informations in the Wikipedia page or in any other page. Okay, so yeah, that's it. So we will meet next week, and from next week the main content of biophotonics course will start, and I hope we will enjoy like biophotonics content during those weeks. So we will get to know lot of interesting things of biophotonics. Okay, so thank you.